So thank you for the invitation. Originally, I was going to present on radiocarbon dating and Bayesian modeling for paleoenvironmental applications using OxCal, but there was, I think, a colleague from Penn State who talked about radiocarbon dating already. So I'm going to breeze through that really fast, but there are aspects of that that really matter when building chronologies, not just in paleoenvironmental applications, but in archaeology as well. So for the things that are really relevant, I'm going to touch upon. But yeah, if you're interested in more radiocarbon dating, I suppose you can go watch that talk on YouTube. So by now you should know what radiocarbon dating is, how it's created, the half-life, and how that allows us to reconstruct or use radiocarbon dating as a clock up to about 55K, how it's dispersed and fixed, the atmospheric and the non-atmospheric reservoirs, so the oceans, rivers, and lakes, and the offsets and the issues that sort of the non-atmospheric reservoirs bring, sort of the challenges, how the calibration curves are built, right, and the different sort of uncertainty and resolution at different time periods, and why calibration is required in the first place, and the radiocarbon dating process, right? So I usually look at it this way, that it's within the black box of radiocarbon dating. We go through the steps that you see in the screen, which are sampling, starting from field sampling, lab sampling, chemical pretreatment. It's when we're essentially trying to decontaminate your samples in the lab. Graphitization, although more and more now labs have the ability to measure your samples from gas, so we don't have to graphitize. AMS measurement, calibration, and then more and more it's becoming part of sort of the radiocarbon or chronometric framework to do Bayesian chronological or age modeling, right? So I'll focus on the last one. But before going through that, I'm going to pause a little bit in some of these steps because it matters later on when you're building your chronologies, specific sort of aspects of the radiocarbon dating process before we get to the Bayesian age modeling. So first, I want to talk about field sampling, which some of my colleagues, sort of radiocarbon dating specialists, will say that field sampling, or sampling to begin with, is the most important step in radiocarbon dating. And this is why, essentially. So in archaeological applications, we often sort of recommend for people to follow what's called water bulks criteria. This was published in 1971, which applies to archaeology, but also paleoenvironmental or paleoecological applications. And that's essentially making sure that we are dating the event of interest by choosing the right material, right? And when we're thinking about it that way, there are two categories that really matter here. One of them is, as you can see on the table, is certainty of association. The second one is thinking about inbuilt age. So for certainty of association, we want to get to that category A, which is full certainty. So in archaeology, this is the archaeological object itself furnishes the measured sample. So human bone from a burial or wood from a house post. So if we're interested in the burial of that human, we're not going to date the wood that's sort of around it, or the charcoal bits, or the sediment, right? We want to date the burial. That's the event of interest. So we're going to go for that material that's going to get us the closest to that event. And then the second thing is inbuilt age. So that just means that the difference between the event of interest and the element that we're dating, the difference in age is so small as to be negligible. So you want to essentially be below sort of, or within uncertainty. So that'd be something like a bone or seeds, or if it's charcoal, something like a twig or something like that, that's sort of very short-lived, like 1 year or something like that. What we don't want is to have the time difference amount to sort of centuries or millennia, right? And so for wood charcoal, which maybe a lot of you are dating, you want to make sure it's not coming from a long-lived species, right? Because some trees can live a very, very long time. So you don't want to be sort of in this category C or potentially D, right? And so this D category is where we don't know that offset, that inbuilt age, especially when it comes to sort of potentially soils that have mixed carbon sources, or organisms living in an aquatic environment, right? So coming off of that atmospheric reservoir. So this is very important for you to keep in mind. And this, again, it's for archaeological applications, but it applies for everyone. So practically, what does that mean? I've just sort of listed a number of materials that are usually dated, and what I want you to get out of this is that I want you to sort of consider and think about whether you're targeting the period of interest, whether it be formation or deposition of soil sediment, and then considering all of the radiocarbon issues, which you are now well aware of, right? From sort of the fast bit I went through, or if you watch the colleague from Penn State, his talk as well, right? So when you're looking at charcoal, make sure that you're dating single entities, so you're not dating multiple charcoal bits, because then you're averaging different carbon sources. Think about inbuilt age, right? So you want to make sure you do species ID, so you identify the species, make sure that it's not something that lived for a very long time, and you're targeting sort of the pith of the tree. And then anything older than 30K, for example, for charcoal, routine pretreatment is not enough. So this is what commercial labs do. It won't be enough for you to decontaminate the sample, and this is something that I'll go through in a second. So I'll leave this there, and then we can share, the talk will be in YouTube, and we can share the slides on PDF as well, so that you can have a look at the other materials. Essentially anything that's aquatic, you have to think about the marine reservoir offset or the freshwater offset. Marine is sort of, we have more data for that, but freshwater is really, it's a bit more difficult, and it's better for you to quantify it in your site and your period as well. So moving on to sort of the next step that I really want us to think about before moving on to Bayesian modeling is chemical pretreatment, is that decontamination of the sample. Modern contamination is more impactful than old or fossil carbon contamination. So if dates are inaccurate, more often than not, they're likely to be age underestimates. So this is a graph showing sort of measured sample age versus actual sample age. If you can see, I can't show my marker, but if you can see that blue line, that black line is no contamination. The blue is an intrusion of 5% fossil or old carbon. So for a sample that's actual age 30,000, you can see there it doesn't make much of a difference. If there's a 5% modern carbon intrusion in your sample of contamination, you can see there for a sample that should be 40,000 years old, it will come up to be, what's that, maybe 23,000. So that's a massive, massive difference. So I always, when I teach my students about radiocarbon dating and pretreatment, for pretreatment really, really matters in two instances. One is if your sample is old, so anything sort of older than 25,000 years old. So if your time period is beyond that, you need to think about this. And then for samples that are really, really small. So small and old, this really, really matters. And we'll talk more about this. So for charcoal, like I said, and for bones as well, anything that's sort of greater than 25 or 30K, these samples or these materials are going to require more rigorous pretreatment. That's often not done at commercial laboratories. So again, you also have to think about sort of where you're sending your samples. So for charcoal, we usually do routine chemistry. That's sort of acid-base acid rinsing. That's enough. If they're older, we have to take it a step beyond and sort of do some sort of combustion, step combustion, wet combustion. That's not routine. And that's necessary again for old carbon or old charcoal. Same for bones, we need ultrafiltration. And you can see here in the study that came out in 2011, what they did is that they took different charcoal samples, the same charcoal, and subjected it to ABA versus that combustion step, so more rigorous methodology. And you can see there in the square in red that the same sample yielded at age of about 34,000 BP when done under ABA, and then 41,650 BP when combusted with that different method. So again, just keep that in mind for your samples. The next thing you should know, so the next step is sort of, when you get your dates from the lab and you sort of start calibrating, I want to focus a little bit more, not about sort of the details of this, but more about results reporting. So this is sort of the table that I usually will give my collaborators, and I'll say this is the table that we need to sort of publish, and we need to make sure that we have all the non-chronometric information. So the context, the material, the species. Again, the species is really important when we're talking about inbuilt age for charcoal, for example, but then chronometric information that you see listed there. So the pretreatment, again, because it has a massive importance on all samples, Again, this is just archaeology, but in 2011, Tom Hyam came out with a paper saying that as many as 70% of the oldest radiocarbon dates in the literature for this time period, so Middle to Upper Paleolithic, may be too young because of modern contamination, essentially. So 70%. This will come up again, so just keep that in mind. Also elemental and stable isotope values. So this information here, so your elemental information, stable isotopes, collagen yield pretreatment, it's what allows us as radiocarbon dating specialists to sort of assess the reliability of your radiocarbon dates. So more and more now, maybe you've encountered it if you're publishing radiocarbon dates and you get a reviewer that's a radiocarbon dating specialist, they will expect you to have this data and they will sort of demand this data. I certainly know some of my colleagues demand it and won't recommend your paper to be published if you don't have this information. So again, this is something that you need to be in touch with your lab about and see if they even do it, if they don't do it, and make sure that you report it if you do have it. This is just the accepted ranges for bone collagen by the Oxford lab, essentially. So what this tells us is that the accepted value, for example, the C to N ratio should be within that range, so 2.9 to 3.5. So if it's higher than that, we know you have more carbon, you have contamination. So this is, again, how we're able to sort of assess reliability. So you need to have this information. And also be minded that a lot of the delta 13 values that the labs will give you are not necessarily IRMS. So if you want to do anything with them, you might not be able to if they're AMS derived because they have fractionation. So keep that in mind. Talk to your lab, essentially. Just very quickly, very basic as well. Make sure you always publish the BP date, not just your calendar or your cal BP date, because we can't easily sort of back engineer your BP date from your cal BP. And if in 5 years there's another calibration curve, people need to use your data, then you need to use your BP or your fraction modern data, not your calibrated BP. The last thing I really want to bang on about is that you need to publish negative results as well. So if you're sending 100 samples to your lab and you get only 50 dates back, that's a really high failure rate. That's unrealistic, maybe not. But that information is useful because it might say that your site sample suffers from poor preservation, for example. So just remember that there's essentially a survival bias with radiocarbon dating, which is also important for something that I'll talk about at the end. And this is just a graph showing the failure rate at the Oxford lab for a number of decades. And something like teeth, bone, charcoal, there is a failure rate. And that is valuable information that I think should be published as well. Just remember, if you're using the marine curve, apply the most appropriate delta R value. There's that really useful database in calib.org, but I find it's not the most up to date. So research your period and place as well. And remember that your delta R changes in time as well. So you need to apply a temporally appropriate delta R, ideally. So finally, I'll just see. Speaking a bit fast because I just want to get to the Bayesian modeling bit. We can talk about Bayesian modeling. So it's Bayesian statistics, essentially, and what that means is that it allows us to sort of incorporate your dates, so these likelihoods, with prior information. So in simpler terms, it means sort of the answer equals dates with the context. So in that sense, context is your prior information. It's what you're assuming is sort of correct information that you're feeding the model. So the most used, at least for me, prior information is ordering, either for archaeological or paleoenvironmental or paleoecological applications. So that could be stratigraphy in archaeology or depth, right? So that's depth is one of the most useful priors for doing age depth models, right? And then also we can use independent temporal markers of known age, like a tephra layer or coins in an archaeological site. So that's your prior information that you're using to feed your model. So in terms of depositional models in OxCal, there's a number of them. I've listed them here. That includes sequence. And I'm using this sort of different font for sort of OxCal code. So anything in that courier font is sort of OxCal code. So first we have sequence when we only know the order information, or that's what's used. A D sequence is something with defined age gaps between events. A U sequence assumes uniform constant deposition. And then a P sequence, which is the one that we usually use when we're doing sort of paleoenvironmental, any lake core record, something like that. That assumes an underlying random Poisson process and allows for fluctuation in deposition rate. So for that fluctuation and that flexibility, we're using the K parameter. I'll go through some of these examples. So a sequence is usually, you'd want to use it for a stratified archaeological site, for example, when you have sort of these layer B is above B, layer A, and then B, and then C, but nothing about rates or gaps are assumed. So you're using only when the relative order is known. And then you have the D sequence. So for example, you'd use that if you have a tree ring sequence where each of the counted rings represents, I don't know, a year, and you can specify fixed intervals between the dated tie points. A U sequence is quite unrealistic, really, but that could be, for example, a Paleothem segment where independent data indicate approximately constant growth rate over the modeled span. So you can use it when it's reasonable to assume that there's a constant uniform accumulation rate between the different boundaries. Again, this is unrealistic, but it might be useful for hypothesis testing. And then finally, again, the one that you are probably using if you've logged in is the P sequence. And that could be a peat bog or a soil profile with episodic inputs and variable accumulation. So you use this when the deposition is monotonic but variable, and you have depths and want an H-depth model with a flexible rate. And that flexibility, again, is controlled by this K parameter. So I'm going to focus a little bit more on the P sequence. If there are questions along the way, just shout. I can't see you, so just unmute yourself and ask a question. So P sequence and the K parameter, what is the K parameter? So it's the assumed granularity of depositional increments per unit depth. So in simpler terms, the K parameter sets how finely OxCal will chop up each centimeter of sediment into depositional steps. So this example here I have on the screen of that would be for like a finely laminated like sediment. That would mean that the model expects about 3 increments per centimeter with the actual number of years per centimeter determined by the dates, right? So that's why you're essentially doing the modeling, right? So you can extrapolate or obtain dates for your depth. So in this graphic, we have a number of the sequences, and just to sort of show you what the K value does. So in black, you have an extremely high K value. So it's entirely uniform and constant, the deposition rate. So that would actually be like a U sequence. And then you have a high value K value in green. So you can sort of see it there, and that represents or reflects a really fine, smooth deposition. And then we have a low K value in blue, and you can see it there in the graph. It's coarser, it's more valuable deposition. And then not plotted, it's an extremely low K value. So that would actually just be a sequence where only sort of the ordering is dictating, right? So you don't sort of know anything about the deposition rate. So how do you estimate the K value so that you can sort of put it in your model? Christopher Bronk-Ramsey in this paper in 2008 and QSR shows this example of how to estimate the K parameter using 3 exposed tephra layers of known age on a profile. So if the spacing is very consistent between the layers and across the sections, the K is really high. Again, a really high K value is just uniform deposition. And then if the spacing varies a lot across the sections between the layers, then you have a really low K value. So then you can essentially sort of use this framework to calculate your own K parameter if you want to input it. However, most of us don't really know what that is, right? That's part of why we're doing sort of a depositional model. So if the K parameter is unknown, you can actually let the model find this K parameter that works best for the data by allowing it to vary over a large range. So this is how you set up sort of a P sequence with this variable K range. So you're again allowing it for the model to identify the K parameter for your sequence. So there you can see P sequence, the name of your sequence. The 1 would be the K value. The 0.5 is the interpolation rate, and then the U there that you can see, that's the K range. So that's a uniform distribution, and you're allowing the K parameter to sort of find itself within that distribution. So building a p-sequence in OxCal, I'm sort of maybe being unfair in assuming that you've used OxCal before. If not, you essentially see what you have there within that sort of first panel on the left of the screen. And the suitable values for a p-sequence are noted there. So you usually enter it sort of based on the units of your depth, so centimeters or meters, and you have the setup there. And for both of those, you'll get an output every 2 centimeters. So that's the k-value divided by the interpolation rate. So if you go to OxCal, you're in that first sort of, I can't show my mouse, but you're in that first tab, that input tab, you hit on tools, models, deposition model, you'll get to this other panel here, deposition model, and you can copy paste your information. So you need your lab code number, the radiocarbon date, your error, and your depth. You can copy paste, put all of your information in there. And if you want a depositional model that's a p-sequence, you just enable that Poisson deposition, so that blue tick check mark that you see there, and it will fill all of that other information based on the depth units that you input in there. If you want to do outlier analysis, which I'll go on to talk about in a second, this is also where you input that. So it will populate everything for you. So if you input that, this is what you essentially get. And then to your left, you can see the model by clicking on that model button essentially, and you see it in that way. If you want to look at the code, you just click on code and you get this. So if you're sharing your code with anyone or you're publishing, this is essentially what you need to publish on your SI, right? So to get the results from a p-sequence, you essentially, as soon as it's done running, you get this table where you have all of your results. And if you want to look at your HDEP model, you're going to hit view from that table tab, and then you can see your HDEP model. And as you can see for this one, this is an imaginary sequence, so it's extremely nice and neat and very ideal, and that's not often the case. But you can see that you have your dates at the top, and then you have a bit of a gap, and this is why the uncertainty is greater, because there's no data feeding the model there. And then your dates at the bottom, essentially. So this is a very nice model and idealistic. If you want to get your ages at Z increments, you essentially have to go from your table tab. You have to hit on that icon with the sort of little, it looks like a little page. And then you get this report. You have to click on depths. And then to your left in that sort of interpolation sedimentation panel, that's where you get your data. You can copy paste it and use it like that, or you can just hit save and it saves it as a CSV file. So then you get your information, and then you can take this to R and do whatever. Although usually if you're using R already, you're not using OxCal, but if you want to get your sedimentation rate plot in OxCal, which is, elsewhere called accumulation plot maybe, you just hit plot in that same screen, and you can see that graph that's to the bottom right there, and you can save it as you wish. I wanted to show you where you can, if you're interested in sort of looking at what the model decided your K value was for this, you essentially just have to you can see the red square there that I've put. You click on the name of your sequence. So in this case, I named it example. So I click on that and you get this graph that plops up. You can see that sort of normal distribution there. You can see your setup of your K parameter. And you can see in this case, your K value is quite positive, is high. And that, again, it's because it's a really nice, almost very uniform deposition rate. So you can see it's positive, right? So it's to the right of 0. If you want to look at what like a low K value looks like, you can see it here. So that's to the left of 0. So that's a more negative value. And you can see there that the deposition rate or the sequence is a lot coarser. So that makes sense. You can save this, put it on your SI, whatever you want as well. So outlier analysis in OxCal, I think the other sort of platforms or programs that you can use in R don't have this capability, and in my opinion, it's one of sort of the biggest advantages of using OxCal. So what is outlier analysis? It essentially addresses the assumption that all the measurements are correct by introducing a prior for each sample being wrong within its context. So that context in relation to the prior framework that you've created. In your case, it might be sort of that age depth ordering, right? So this is something very reasonable to do because we know that dates are not necessarily correct all the time. So assuming that they are just by default correct, it's not necessarily um, a fair enough assumption, if you see what I mean. Because, we might have pretreatment issues, vertical mixing, taphonomy issues, and whatnot. So the advantages of this is that it will identify objectively your outliers rather than sort of relying on manual exclusion. I'm a radiocarbon dating scientist and I work with archaeologists and paleoecologists and everyone across the board, sometimes when you give them dates that they don't like, that they don't think that they're either too early or too young, they might not want to publish them or might not definitely include them in a Bayesian or an age model, which is not the transparent and the objective way to go, right? So at least using outlier analysis allows you to sort of objectively identify those outliers. You don't have to do that manual exclusion, which I'm not a fan of. So then after it identifies the outliers, it will downweigh them according to their posterior. So if it identifies that one age is an outlier at 95%, it will essentially mostly discard it from the model, right? So it won't appear, it won't be taken into account in 95% of the iterations that the model runs. And that's really useful. So the typical assumptions here for example, the general outlier model, which is the one I use the most, is that 95% of the measurements are correct and correspond within that context, with 5% being wrong or at least dating somewhere near the context. So for the general outlier model, the offset of the true date in the measurement is drawn using a student's t-distribution. So you can see it in the little graph at the bottom there in the orange, that's your student's t distribution, which is just a normal distribution that's a bit sort of broader at the end. And so what this means is that your outlier can be sort of too young and too old, right? So it's on either side of zero. You might want to use the charcoal outlier analysis in OxCal, which essentially, you're essentially telling it that every charcoal date is too old. There's inbuilt age in that measurement. So there you can see essentially that it's sort of to the left of zero. So it can only be too old. Okay, but if you use sort of the general outlier, the charcoal, OxCal has these defaults and it will sort of populate everything for you. You can make your own sort of outlier analysis, but there's definitely applications where you can do that, but often people will use one of these default models within OxCal, with general outlier being sort of the most flexible and the one I often use because again, your outlier can be too young or too old, which is quite reasonable and realistic. So how do you set up the outlier model? If you go to build your depositional model, like I showed you before, you have that option to add your outlier model. If you click on general, it will sort of fill everything in based on what the general model usually does. So you don't really have to do anything. And you can follow how to do that. I've put sort of the instructions you can see in red in every slide. So if you want to go off and play with this, you can hopefully follow from there. So what does that look like? So you can see there in the code, it adds it to the top above your sequence. And then with each date, you essentially also specify that prior probability of it being an outlier. So in all of these cases, just uniformly, I'm applying a 5% probability that they are wrong, essentially. How do you look at the results? When you look at your table, which is your results, you can go on view, outlier, and then you get this report that you see at the right of the screen, where you have the prior and the posterior probability for each date, and that's the result of the outlier analysis. So all of them, you can see the prior is 5 because that's what I set to begin with, and then your posterior, that's your results, right? So in all of them, most of them are green, that's really nice, but one of them is 100% outlier. So it's identified as 100% and it's discounted by the model essentially. So again, you can leave in dates that are outliers and you can leave the model to essentially remove them from the model. So it's, again, very transparent. Just a little note, if you're using outlier model, don't report the results for that and your agreement indices. I find that a lot in the literature. So the latter functions by assuming that all the determinations contribute at full weight. So once the outlier model will adjust these weights, the statistical basis for sort of the agreement indices no longer holds, right? So if you're using an outlier model in OxCal, just report that. Um, the results again, you can choose to sort of, I guess, publish that little report with the green and the red, but I think the best way to do it is to plot it. So when you're showing your age-step model, you can plot the results of your outlier model in the plot. So you can see there it's listed under each age. You have that O, which is outlier. You have your posterior, so in that case is 2%. And your prior. So all of them, again, it's going to be 5%. So none of the ones that you can see in the plot there are identified as major outliers, which is usually we do sort of anything greater than 60%. So for that, I've shown you how to do it there. You go to the plot, edit format, and then you enable that outlier sort of section there. Or that line, and then you'll see it plotted in this way. And so that way, when you're talking about the results of the outlier analysis in your text, you can refer to the figure, and there you have the results. Some useful OxCal functions capabilities. One of the really useful things is obtaining a date for an undated event. For this, you can use the date function. So here we have a sequence. So it's a core where you have your depth, your dates, and then you're interested in event A. That's the red square essentially. And you have an undated event that has a particular depth. So there you can see it in the model form and then in your code how it looks like. So here you can go to the results and you can see the date for that event that's not dated. So if you click on your event A and that's how you named it, you'll get the OxCal plot for it. And there you have it listed or noted at a 95% probability. And you can save that. You can do whatever you like with that. You also get the results on the table as well. Another really, really useful capability of OxCal is the difference command. So here I had a typo I forgot to take out, but essentially what you can see there is that I have two adjacent cores. So they're sort of like this in the same plot. That's meant to say same. And you want to essentially see how comparable two events. So you have two events in the core that you think it's exactly the same event, essentially. You want to see how they compare statistically if they're dating to the same time period. So you enter it like that. So it's the difference command, the name of that difference that you're trying to estimate, your event A and your event B. So that's how it looks like. And again, you're just testing whether the age of these two events are comparable between these two separate cores or adjacent cores in this case. So for this, again, you can just click on the name of the difference command, that whatever you gave it, and you can look at the results. So in this case, for example, the two events, the age between them is comparable because the results include 0. So 5 minus 5 is 0, right? So it means that there's no difference, right? So if it doesn't include 0, then you have the events or whatever the distribution is sort of statistically incomparable. So this is something really useful that I use a lot when I do sensitivity testing, and I'll go into this in a bit. Another thing that you can do is cross-referencing. So again, hopefully you can sort of do this on your own as well, or it might be useful to you. But again, I have these two adjacent cores in the same plot. I have a tephra age that I entered in core 1. You can see there, it's called tephra 2, and it's a normal distribution, might be an argon-argon date. And then I have the same tephra cross-referenced in core number 2. So the way that you do that is that you have to write it with that sort of equal symbol and then the name of that tephra. So it has to match exactly the name, and you have to preface it with the equal symbol or sign within the quotation marks. So you're cross-referencing things, and it's quite useful for different applications as well. Another thing that you might be interested in if you're using OxCal is estimating the duration of a specific event. So for that, you would want to use the interval function. So in here, we have two sequential cores in the same plot, right? So we have a base core, and then we have a gap, and then we have a more sort of the core that's at the top essentially, right? So you enter this sort of interval command that you can see there, and it's showing sort of in blue on the left, and that's how it looks like in the code. And here you can again click on the results, or you can see the results on the table. And essentially here, what it's saying is that the duration of the gap between the two separate cores is between 760 to 2,660 years at a CI of 95%. And you can change the confidence interval. You can tell it to quote it at 68% if you want, if the distribution is normal. You can also, if you have a lot of gaps, if you have many cores, or if you have a lot of interval functions that you've put in, you can plot all of them by hitting on the table tab, you go view, plot intervals, and it'll plot all of your intervals, right? So here I'm just plotting the one, but you can do that as well. That's quite useful. Two little things that I wanted to show you is that you can actually incorporate an age within a boundary. So if you know one of the boundaries in your core, it has an independent measurement that you're absolutely sure that's the age of your boundary. You can see it incorporated there. So it's a normal distribution, top A core. You can incorporate it. So this is an example, but you can also incorporate a modern age for the top of your core if it's applicable. So this would be sort of that uniform or that top hat distribution that I have listed there. You can also change the resolution in OxCal. So the default is 5 years, but for some reason you might want to change that. So I've changed this here at the top to 200, and you can just use the same code that I have there if you want to copy this. Or if you're doing any bomb stuff where the resolution is a lot less than 5 years, then you're going to decrease that resolution, and this is the place to do it. Combining ages, I want to quickly check. If you're wanting to average ages, you're going to use rcombine or combine. So rcombine, it has the R, so it's for radiocarbon dates. And then combine is to average replicate calendar ages. I really emphasize on replicates there, and I define replicates as samples from within the same time window. So it would be a duplicate, a replicate, or one that's short compared to measurement uncertainty. So by a factor of 10. So if you have an error of 50 years, it's 5 years. If you have multiple dates, but they're not replicates, but you're trying to sort of date the age of that particular event, and that is just represented by an age on a charcoal, bone, pollen, whatever, you're better off doing sort of a single face and putting that date function that I showed you earlier, and that you'll get a date for that event that you're interested in. So you're not averaging dates that are not necessarily representing replicates or duplicates. That would be a bit more maybe inappropriate. If you do any of these, so combine, it runs a chi-square test. So you can click on it like I've done there, and you can see the results of that test. Another thing that people really like doing, and I'm going to talk a little bit more about this, is summarizing ages. So for that, you can use a couple of options in OxCal. The one that is used the most after a paper that sort of Christopher put out just on this, so you can look at that, Bronk-Ramsey 2017, I believe. It's just on summarizing ages. It's the KDE model, so you can see it represented there. You can insert it by sort of following the instructions on the screen, and that's sort of a method that's a statistically robust, smooth estimate. It will give you a smooth estimate of the true age distribution and avoiding artifacts from calibration. So it uses MCMC to find the underlying distribution. So for those of you who have never used sort of a KDE model or done anything like that, it's essentially a very sophisticated histogram, right? So you're getting the frequency of your chronometric data. And this is how you enter it. So you can have a look here if you want to do that for yourself. So it's just a KDE model function, and you put all of your dates within that. When you're looking at the results, you can click on whatever you named your KDE. So there's that red square that I have there, and then you'll get this plot. So that's your KDE. The sort of very maybe like noisy gray distribution that's in the background, that's the result of some distribution. The one at the front, so that darker gray, that's your KDE, and the blue is sort of your uncertainty envelopes. So you can look at that. When people do KDEs, though, I do recommend that you, if you're plotting or if you're showing this figure, I think you should add your sample size, because like KDE of 1,000 dates is not the same as a KDE of 10 dates, right? So the reader or the reviewer needs to know the sample size. Also for OxCal, you can just get it to plot the calibration curve in the background as well, which is quite useful. You can do a rug plot to look at sort of, you'll see where your measurements lie in the x-axis. So there's a number of ways to play with that plot for publication. Another thing I just wanted to show you is that if you're, so for example, in Brazil or somewhere else where you're maybe in the southern hemisphere, but you get influence northern air masses, so you might want to combine your atmospheric curves. You can do that in OxCal as well. So you can see there on the right, I'm calling to the model in Cal 20, SH Cal 20, and then I'm doing a mixed curve with a uniform distribution. So it's going to pick from both essentially. It is going to yield sort of your estimates are going to be, the uncertainty is going to grow because you have sort of this added uncertainty here. The other thing that you can do, which is I really, really recommend, is sensitivity testing. So you can run a model A using only in Cal 20, and then a model B using SHCal, and then test the difference using a difference command like I showed you for a specific event or specific events. So I always say that, or I recommend sensitivity testing, right? So you can see whether your different priors have an impact on your posteriors. And all of that sensitivity testing is really good, it's really robust, and you can just include it on your SI. A couple more if you're interested. So one of my colleagues, for example, might use OxCal but wants to add age uncertainty. So for change point analysis, for example, so for that, you essentially need to extract all of or a number of ensembles. So that's a stored set of MCMC iterations from OxCal. This is how you do it. So if you want to extract ensembles for use in R, you essentially have to add this option and you give it the number of ensembles that you want, like you see on the screen. And then you can plot it so you can see every sort of black line is a different sort of model iteration. It should be 500 in here as we specified. And then you can extract that data as well in the same way that we did before, essentially. So you can see there in the red square, you can see the headings. So you'll have a date essentially for all of these different ensembles per depth, right? So you can save that or you can copy and paste that and then use that in R however you wish to sort of incorporate uncertainty. I also wanted to show you that you can also save all of that data that's in that table, in that results table. If you just go on that table, file, export, save, this is what you're getting. It's a whole lot of data because you're getting data points for every sort of probability of each age, for example. So the way I've used this, and just an example, if I can click on it, yeah. I've taken this data in the past when I'm plotting radiocarbon dates or modeled ages. I don't want the dots to be static, essentially. I want them to represent the radiocarbon age as a distribution because that's what it is. So essentially in R, you can get the size of your circle in this case to be conditioned on the probability. So if you have a date that has a unimodal distribution, so a normal distribution, your circle would be small at the beginning, would grow with the distribution, and then shrink, right? So I think it's a much more sort of appropriate way of plotting chronometric data like this. So you can get that out of OxCal and then use it in R. Lastly, a couple more things on this, just extra sort of tips. You can annotate OxCal using the two sort of forward slashes. You can add information tags to each date. You can see there in that R date, I have longitude, latitude, the Delta 13C material, so that's text and quotation context. You can also, which is how I build Bayesian models, you can actually just get Excel to write your OxCal code using an equation. There's a fast way to out a lot of data with information. And if you add that information, those tags, you can, for example, get OxCal. You can map things in OxCal. So I assume that all of you will have like an Excel sheet where you have your radiocarbon date, the lab code, the date, the error, and maybe your depth, and I don't know, you can add more stuff, your material, your species, blah blah blah. So you can easily just get, again, using sort of that I've put there to get OxCal to write you or Excel to write you your OxCal code. It's the best way of using OxCal, I think. You can also increase the K iteration, so that's an option. That's default to 30,000. For complex or inconsistent sequences. So you have it there. And what's essentially, that's what you're doing. So if you double it, so if you set it to 60,000, is that you're giving the model more time, more space to sort of reach convergence or to find the solution before it sort of quits. It will take longer because you've doubled the iterations that you're asking it to do, but you can do that as well. And then you can do mapping. I don't know if a lot of people know you can do this, but you can map in OxCal. So in this case, I have all of my radiocarbon dates tagged with the delta 13C. So on the table, you can go view, plot on map, and then you can split it according to your, I don't know, stable isotope values if you want. And then you get, what you see that map on the right, and then you can actually just hit play, and it's sort of like a little movie. You can do this sort of, I use it as maybe like a first order way of visualizing data. I tend to like to just take it to R and create maps that way, but I show it in case it's useful to you. Finally, on an archaeological note, I know a lot of folks out there, sort of paleoenvironmental folks, um, if they're doing an analysis where they're sort of correlating human environment climate dynamics, what they take for human from the archaeological record as a proxy might be sort of the frequency of dates. So they summarize distributions to compare to the paleoenvironmental data and whatnot. I just want you to know that it's tricky and it's not very straightforward, and it often I think shouldn't be done. So here I just have using the frequency of dates to surmise population density or demography. So that's how it's usually done. So the thing is here that the more radiocarbon dates, there's a rise in population, right? And so this is what folks are usually correlating to their paleoenvironmental evidence. I don't know, changes in vegetation or fire regime. So this is what it would look like. This is a plot. So these are radiocarbon ages for the middle to upper Paleolithic transition in Iberia. If you look at the gray KDE, you might sort of conclude that there's an increase in population at around 34,000 years ago in Iberia, right? So that you get the biggest sort of peak in the distribution. However, and now you guys know all about this, this is not the case. Why? So when was this? 2020, we put out a little paper talking about the challenges of sort of doing these types of numerical studies. So they're generally called radiocarbon dates as data studies from a radiocarbon dating specialist perspective. And so you guys know by now that anything that's older than sort of 25K, especially for bone collagen, charcoal as well, if you use the wrong methodology, you're getting very different results. And so it will be a lot sort of younger. So here, if we look at that distribution again in that graph, the gray actually represents the data set before ultrafiltration. So that's that rigorous methodology, and blue is after. So actually, that gray distribution is wrong. So it's including measurements that are really, really inaccurate. So once you see the blue distribution, that supposed peak in population density is no longer there. So it's an artifact of inefficient pretreatment methodology. So there in red, I have an increase in population at 34K in Iberia. No, it's just inaccurate ages, confusing interpretation. So remember this if your period is sort of greater than 25K. So if you're looking at Europe, Australia, and you're using radiocarbon dates from archaeological settings in this way, so you're just summarizing them and correlating them to your paleoenvironmental records, warning, right? There's a lot of issues with it. So apart from the inclusion of inaccurate ages, these type of numerical approaches also take the ages out of context, right? So in archaeology, when we're building chronologies, I advocate for it, and a lot of my colleagues do, in that Bayesian chronological modeling is a more robust approach in chronology building, right? So we always, well, I teach that Bayesian chronological modeling in archaeology allows us to do three things: integrate archaeological and chronometric data, which is key; two, statistically evaluate sequences; and then three, correlate multiple sites spatial temporally. So in the middle, we have an archaeological sequence, and in the inside of the red square, you have a number of outliers, right? And I think it turns out here that at that point, it's a cemetery. So these ages are outliers, and it makes sense. So outliers in this sense are really informative, right? So they're indicating pretreatment issues, vertical missing, etc. If you're not doing this though, but you're including all of the ages in a summarized distribution, these outliers are potentially confounding. And then it's not just clear outliers like this that maybe authors might be like, this is a clear outlier. It's not just that, right? So just be really careful with that. Finally, I'm getting to the end. If you need help, look through the literature for OxCal code. People have done maybe what you're trying to do in the past. Join the OxCal group. Someone's probably asked the same question that you have in your mind, and Christopher and other really nice and competent people are constantly replying, and it's a really friendly community. Ask your local radiocarbon expert, ideally a research lab. There's a lot of issues with commercial facilities. Some of them I already discussed, especially if it comes to sort of pre-treatment methodology. And then finally, I'm just plugging the short course in OxCal, essentially, that's run in Oxford and where Christopher teaches. And that happens every year in March. And I just checked with Christopher, and those should be the dates, 16th to 18th of March. You do have to pay, which sucks. There is a hybrid option, so you can do it online from the comfort of your home as well. Sorry. And then just announcements. There's three massive radiocarbon chronologies in Latin America. If you're interested, they're happening back to back for a reason. The first one is radiocarbon archaeology. You might not bite into that, but put it in there anyways. And then we have radiocarbon and diet. And then lastly, it's the third Latin American radiocarbon conference in Santiago. And that's everything. So archaeology, paleoenvironment, carbon cycle, forensics, anything related to radiocarbon. It's a really nice community. So I encourage you to come to Latin America next year. It's where it's all happening in radiocarbon. Thank you. And if you have any questions, let me know. And thanks to Alejandra and the team for the invitation.
Thank you very much, Lorena. So we have time for a few questions. So if you have, like to ask a question, either you can raise your hand or you can also write it down on the Q&A section, and then we will read it for you. Let's see. So in the meantime, I do have a question. So one about like age reversals in sediment records, which can be very common. So
Yeah.
how do you recommend to treat them?
Age reversals. I mean, what's gonna end up happening, for example, if you're using OxCal and you're doing outlier analysis, is that it's gonna find an issue, it's gonna identify outliers, and then essentially your uncertainty is gonna grow. So I think from a radiocarbon dating perspective, when I've worked with folks in doing lake course or whatever, if there are any issues in the sequence, you go back and increase the dating. So if you're interested in any particular part of your core, or if there are issues, the only way of sort of fixing it is by increasing resolution. If you have an age reversal, there's nothing you can do. It is what it is, and you're going to have increased uncertainty in that specific part of your sequence. Ideally, you can go back to your collaborator or the lab and then do a little bit more dating to elicit, shed light on that specific part of your core. But it is what it is.
Yeah, thank you.
I just want to
Any,
say also, just
yeah.
if that happens, what did you date? So I wanted to just also note, if you're sending sediment to labs,
Mhm.
your labs are dating different chemical fractions. So if you send it to, I don't know, Beta, I think they do an acid wash, so you're getting a bulk organic. If you send it to Oxford, they're giving you the alkali insoluble. They're different chemical fractions. So a lot of the collaborators I've worked with, paleoecologists, don't realize that they're actually getting dates for different chemical fractions within their sediment. So often it might be that you're getting issues within your core. It's because it's different materials and you're dating different events entirely. So think about what you dated as well and what the lab gave you and what they dated, because we can
Oh, okay.
target different things.
Yeah. Thank you very much. I think Sarah has a question.
Yeah, thanks, Lorena. That was really interesting. And I guess I wanted to say that I really appreciate that you kind of like the approach of objectively determining the outliers, publishing everything, and then also that the idea that an outlier isn't just bad data, but that it can be really informative. And so I guess, but like when you see your first like age model and you see the dates like not lining up perfectly, it can be really frustrating right away.
Yeah.
And when you realize you have outliers, it sounds really bad. So I guess like what do you find are like the most common sources of outliers and like what's your approach to trying to figure out what's happening?
Um, I think often when I've worked with folks and we get a sequence where there's a lot of outliers, everyone panics. Um, and I've had it where they go, well, did you do something wrong in the lab? And it's like, no, we have standards. It's not, this is real. And then often they go back to their data and they'll look at their elemental data, whatever analysis they did, and they go, oh, fine, actually, we can explain this based on the pollen, the elements, whatever analysis they did, eye tracks, whatever, that there's some sort of taphonomic event, there's an erosion event, there's whatever. So I think it freaks people out, but it is what it is, and it's informative. Um, so I think sort of taking a breath and then analyzing absolutely everything. If you have a record where you dated, everything is really consistent and it's a single material, then I guess that's good. You can rule out dating different events because every material is a different type of event. So that's good. And then you jump onto sort of your data and see what your data can explain. Um, I don't know. I think it's a very case by case basis, but I think the important thing is that it's informative and not to sort of freak out. And at the end of the day, if, yeah, you might have more increased uncertainty and it's not so neat. But again, if you have funding and if you want to resolve anything, then you try to get more dating. Um, but then you need funding for that. So it's very tricky.
So I'm hearing you say don't blame the lab, it's probably not the lab.
Sometimes it's the lab, right? Because what I'm
Yeah.
saying is that if your samples are really old and you've sent it to a commercial lab, your charcoal age is probably wrong.
Yeah.
And that's, if you look at the record in Australia, for example, that's where this all started. So a lot of the ages in Australia are off, wrong. And so, yeah, sometimes the labs are wrong because there's been advances in methodology. So that it can be, I always say it's, you have outliers, pretreatment issues, taphonomy, vertical mixing, a number of things.
Thanks.
Thank you. Anyone else? Um, yeah, I don't see any hands. So, well, thank you very much again. Hello, Lorena. This was great. So Lorena has agreed, like she's gonna share with us the PDF of her PowerPoint presentation, and we're gonna share it with you all, so you can also take a closer look, and then even try to replicate some of the analysis that Lorena showed us today. So with that, just a reminder, next month we're gonna have another talk, so please stay tuned. And if you have any questions or suggestions for future workshops, also you can always contact us. Thank you, everyone.